pharma setups like in diagnostics, right? So sensor word we could really understand that like something which sense the reaction and like then it converts into signal that can really be understood by human, right? Or to provide a data, whether sometimes it is a qualitative in nature and sometimes maybe it is in the quantitative terminologies. Now, how biosensor terms comes into the pictures? Like if you see the sensor, like sensor has mainly two components, right? The one is the sensing element and second one is the transducers, which really converts those reactions between sensing elements with the analyte, right? And that can be displayed and can be, really understood, right? So now in case of biosensors, we have obviously two parts because it's anyways a sensor, but like the detecting device or sensing element is kind of from biological components, and that's why we are calling it as a bioreceptor, right? And the transducers will remains the same, like which will sense the reaction between your bioreceptor and your analyte, right? And will provide qualitative or quantitative uh, data with respect to the reaction happens between these two components, right? So biomolecules may be an enzyme or antibodies or maybe a receptors or organelles. Sometimes it may be a microorganism as well as plant and animal cells, sometimes tissues, right? So there are vast majority of the biomolecules which are being used as an bioreceptor for the development of biosensors, right? And the simplest example is like if you consider the pregnancy detection kit or your blood glucose level detectors which are nowadays available, right? So these are the simplest form of biosensors, right? And that help us to diagnose certain elements, right? So this is the pictorial representation, right? So as I mentioned, the NIT, like for which you wanted to have a detection system, right? So if we consider the glucose, right? So here, like glucose, we can consider as an analyte.